So today we have two trainee talks that will be given. The first person I'll introduce is actually going to be the second person giving the talk today, but his name is Nathan Mohar. He is a current PhD candidate in the lab of Dr. Wallrath at the University of Iowa, and his dissertation topic is focused on genetic modifiers and treatments for striated muscle laminopathies. Nathan received his PhD from The Ohio State University in May of 2021. And while he was a student there, he had undergraduate research experience in the lab of Dr. Harold Fisk. And he researched the degradation of MPS1 protein kinase as a regulator of centrosome duplication and aneuploid division in tumorigenesis. In his PhD so far, he has had many accomplishments, one of which is that he received a very competitive award um, from the American Heart Association as as the predoctoral fellowship. And he was also on the T32 in pharmacological sciences within his institution. So far, Nathan has two first author publications, one of which is in revisions and will be out soon. But we're really excited to hear his talk today and excited to learn more about his research.
Okay. Thank you to the organizers for giving me the chance to present today. As I said at the beginning, I'm Nathan Mohar. I'm a fourth year genetics PhD candidate in Lori Wallrath's lab at the University of Iowa, and I will be talking about genetic modifiers and treatments for striated muscle laminopathies. Maybe there we go. So kind of the question that I aim to answer with my research is that many monogenic diseases still result in variable phenotypes. So as most people are probably aware, monogenic diseases means that a mutation in a single gene causes disease, and these are inherited as autosomal dominant, autosomal recessive, or linked to either of the sex chromosomes. However, many monogenic diseases present with a spectrum of disease onset and disease severity that kind of goes against the relationship to only a single genetic factor. So some of this variability can be attributed to genetic modifiers, which are differences in the genetic background of affected individuals that can alter the phenotypes associated with monogenic disease and either increase severity in the case of an enhancer or decrease severity in the case of a suppressor. So I will be, for my thesis, I study genetic modifiers through the lens of the element A gene. So element A encodes the A-type lamins, lamin A and lamin C via alternative splicing. Lamins are intermediate filaments, which contribute to the meshwork lining the internuclear membrane, known as the nuclear lamina. And the structures of both the gene and protein are shown here. The nuclear lamina has many functions and interactors that help mediate those functions. It interacts with the link complex and ultimately the cytoskeleton to mediate the nuclear response to mechanical stress. It regulates nuclear import and export by stabilizing nuclear pores, directly interacts with chromatin to regulate gene expression, and is also involved in several signaling pathways. Shifting the focus back to disease, mutations in element A cause laminopathies. Mutations in element A cause at least 16 clinically distinct phenotypes, which are broadly binned into peripheral neuropathy, early onset aging, or progeria, striated muscle disease, disease of adipose tissue, or lipodystrophy. There's over 500 pathogenic mutations reported, and these are spread across the full length of the gene, and there are some proposed but ultimately limited genotype phenotype correlations in terms of mutations in specific regions of the protein only being associated with a specific type of disease. And the mutations are typically inherited in an autosomal dominant fashion. I particularly study the element A-associated muscular dystrophies, collectively known as element A-MD, which are the most common laminopathies. Element A-MD further breaks down into three subtypes: limb-girdle muscular dystrophy type 1b, Emery-Dreifuss muscular dystrophy type 2, and lamin-associated congenital muscular dystrophy, all three of which often co-occur with cardiac defects, most commonly dilated cardiomyopathy that is arrhythmogenic. Specifically, my research question focuses on the fact that individuals with the same element A mutation often have completely different clinical diagnoses, whether it's by broader disease bin or particular type of striated muscle disease. And then Furthermore, individuals with the same clinical diagnosis have highly variable phenotypic severity in terms of things like age of onset, rate of disease progression, and ultimate endpoint of disease, which is typically loss of ambulation. And this variability exists especially among siblings. So this variability has long been attributed to genetic modifiers, but limited identification of these modifiers has occurred to date. So the long-term goal of my research is to understand the genetic differences underlying the phenotypic variability in LMNA-associated muscle disease and ultimately use these genetic differences to identify candidate treatments for the disease. How I have gone about that is through single family studies of siblings with variable disease phenotypes. So, one example of that is this family shown on the pedigree here, in which four siblings inherited the same LMNA mutation causing the amino acid substitution R527P originally from their father. Their father was mildly affected. He did not lose ambulation until his late 50s before ultimately passing away from cardiac complications of the disease. Unlike their father, which they originally inherited the mutation from, two of his offspring that are now in the second generation have very severe disease. They were both diagnosed in childhood. They lost ambulation in their early 30s and have very severe joint contractures that were not present in the father. On the flip side are the other two siblings, which have very mild disease. They are both past the age that their two siblings lost ambulation and remain fully ambulatory and physically active and have a disease presentation that mimics that of the father. So this is then suggestive of the fact that there was a maternally inherited modifier driving the more severe disease presentation in two of the four siblings. And to identify what that modifier is, we did whole genome sequencing in collaboration with Dr. Ben Darborough at the University of Iowa. We whole genome sequenced all four siblings and all four affected siblings and look for variants present in both of the severely affected individuals and absent in both of the mildly affected individuals. This ultimately, after prioritization, this ultimately came up with variants of uncertain significance in 24 genes that fit that segregation pattern, of which we focused on this variant in SMAD7 for our further analysis. Reason why we focused on SMAD7 is because SMAD7 is the negative regulator of the SMAD signaling pathway. The SMAD signaling pathway is downstream of both the TGF beta and BMP receptors, and overactivation of the TGF beta pathway in particular is heavily implicated as a modifying factor in other forms of muscular dystrophy. So our hypothesis is that if you have a loss of function variant in SMAD7, you essentially cut the brakes on this pathway and lead to overactive signaling, and that variation in SMAD7 could modulate the severity of LMNA-associated muscular dystrophy and ultimately cause the phenotypic difference among the four siblings. So to isolate the effect of this modifier, we turned to modeling in vivo modeling in Drosophila. So the LMNA gene in humans is orthologous to the LAMC gene in Drosophila, and in the Wallrath Lab, we have a long history of making these transgenic Drosophila models of orthologous LMNA mutations that we can then express specifically in muscle using the Gal4-UAS tissue-specific expression system and generate offspring that we can use to study the effect of the LMNA mutations. So in this case, we made flies that are transgenic for the corresponding R564P mutation, which corresponds to R527P. To model the SMAD7 variant, we turned to CRISPR. SMAD7 is orthologous to the DAD in flies. We edited the endogenous DAD gene to contain the corresponding variant to what was present in the family. And then we ultimately combined these into a single model where we can express the mutant lamin either with or without the SMAD7 variant in the genetic background, with the rest of the genetic background being otherwise completely identical. to isolate specifically the effect of the SMAD7 variant on phenotypes caused by expression of the mutant laminin. So to do this, we turn to various phenotypic assays, first of which we're expressing mutant lamin in the larval body wall muscle and ultimately decreasing the locomotor ability of the flies, which we have published previously with as an effect of several element A mutations. In this particular case, can see qualitatively on the left and quantified through two separate metrics on the right that the element A mutation shown in green causes a severe effect on its own, consistent with the fact that it is individually pathogenic. The SMAD7 mutation alone, shown in blue, causes a very mild effect, consistent with the action of a genetic modifier. However, in combination, the two variants together result in the most severe loss of locomotor ability phenotype, indicating that in this particular assay, the SMAD7 variant is able to enhance the phenotypic defect caused by mutant lamins. To make sure that this wasn't either assay or muscle type specific, we turned to another assay in flies, where in this case, we're expressing mutant lamins in what's called the indirect flight muscle. The indirect flight muscle is indispensable for life, so we're actually able to age these flies into adulthood. And when we express mutant lamins in the indirect flight muscle, we get these various wing posturing defects that are an indication of loss of function of that muscle, preventing the flies from basically holding their wings where they should be. However, these phenotypes are not 100% penetrant, so we can use the frequency of these phenotypes as a readout for degree of loss of function of this muscle on a population basis. And in this case, we again show the same trend in our findings that in both males and females, the mutant lamin alone, shown again in green, leads to a significant amount of wing posturing defects. However, this phenotype is again enhanced by the addition of the SMAD7 or DAD in flies variant to the genetic background shown in red. So further supporting our previous data now in a different muscle type and in a different assay and in a different developmental stage that this SMAD7 or DAD variant is able to enhance phenotypes caused by mutant lamins. So next we turn to trying to address mechanism. So the SMAD pathway in humans is highly conserved in flies. It was actually originally discovered in flies. And in the case of pathway activation, whether it be through TGF beta or BMP, the positive regulators of the pathway are phosphorylated and ultimately translocate into the nucleus where they regulate gene expression. So we sought to assess this nuclear translocation as a readout for pathway activity and determine the activity states of this pathway in our various models. When we do this, I have quantification for this on the next slide, but I think it's fairly easy to appreciate qualitatively that we get an increase in SMAD pathway activation left to right on this image shown here is nuclear staining. So when we overexpress wild type lamin as a control, we get very little concerted nuclear staining. The SMAD7 variant on its own also, you start to see some nuclear staining, but again, it results in very little, consistent with the fact that it is not individually pathogenic. The mutant lamin activates the SMAD pathway on its own, which was a finding we were very excited about, consistent with the fact that it's able to drive disease on its own. However, the mutant lamin in combination with the SMAD7 and DAD variant does lead to the most pathway activation, again shown here as nuclear translocation of the positive regulators, and that is quantified here. So this is then supporting, as this suggests a mechanism supporting our phenotype that activation of this pathway correlate, or activation state and activation level of this pathway correlates with the level of disease severity we're seeing in our models in both of our phenotypic assays. So then to further confirm this mechanism, we said if you can introduce variants into SMAD7 or DAD and enhance this pathway activation and enhance our muscle phenotypes, can you overexpress wild type SMAD7 or DAD, effectively shut the signaling pathway down, and ultimately rescue our phenotypic defects? And we showed that that was possible. So looking here at larval locomotor ability again, looking at these three genotypes on the right, the mutant lamin alone severely decreases larval locomotor ability. The combination of the two variants decreases it more. And then the mutant lamin alone plus overexpression of wild type Drosophila SMAD7 very robustly rescues our larval crawling defect, which is further quantified here, suggesting ultimately that the SMAD pathway, both helping to confirm our mechanism and suggesting ultimately that the SMAD pathway may be a potential therapeutic target in this disease, given that the rescue is to near wild type levels. So then moving forward from this, we started in humans with all of the variant identification and coming up with the basis of the study, and then turned to functional assays in flies to assess the effect of the variants. And now we wanted to translate our findings back to humans. So one of our primary findings was that the mutant lamin alone activates the SMAD signaling pathway, which creates an initial level of pathway activation that is then enhanceable by the addition of the modifier variant, which does very little to activate the pathway on its own if that initial activation doesn't exist. So we sought to determine, oh, actually, sorry, before that, we quantified the nuclear staining in flies again and showed that our SMAD7 overexpression is shutting this pathway down. So sorry, now switching gears to human, we completed the same nuclear staining in human muscle biopsy tissue from three individuals that all are unrelated and have different element A mutations, both from each other and from the family originally under study, and showed that in these three individuals, compared to an age-matched control, there was also significant nuclear staining of the phosphorylated R-SMADs and significant activation of this pathway in the muscle of these individuals. And this is quantified on this slide as well, further suggesting that in people, this pathway activation does exist as it does in our fly model as a result of element A mutations, with this initial level of pathway activation ultimately being necessary for a modifier variant to enhance the pathway activation of this SMAD pathway and ultimately enhance disease. So that brings us to these conclusions that mutant lamins activate SMAD signaling in Drosophila and human muscle. The SMAD7's variant under investigation, modeled in the DAD gene in flies, enhances phenotypic defects caused by mutant lamins through a further upregulation of SMAD signaling. Overexpression of wild type SMAD7 or DAD rescues muscle defects caused by mutant lamins through a corresponding reduction in that SMAD signaling, and the SMAD pathway offers new therapeutic targets for element A MD, ultimately leading us to this model where when you have obviously a wild type element A gene, you do not have an abnormal level of SMAD activity and do not have disease. When you have an element A mutation on its own, you have some SMAD activity that is sufficient to drive mild disease symptoms. However, when you add in the SMAD7 variant to the genetic background, you then further increase the level of SMAD activity, ultimately resulting in severe EDMD in the case of the disease the humans in this study are diagnosed with. So with that, I'd like to acknowledge both the current and former members of my lab, this family, for ultimately donating their information to the study, our collaborators at the University of Iowa, and our funding sources. And with that, I will take any questions.